Welcome, everyone, to this episode of Currently Speaking, the podcast of the Current Consulting Group. I'm Bill Current, the president and founder of the Current Consulting Group, and I'm joined today by Brian Feeley, vice president of the Current Consulting Group. Welcome, Brian.
Well, thank you, Bill.
Brian, give us a little background on your career in the drug testing industry.
Sure. So I spent quite a few years kind of in the lab at the bench developing products. I was in R&D for quite a few years. And I kind of got interested in the sales and marketing side of things. And so from research and lab work, I kind of transitioned into business development and sales and marketing. And then ended up at OraSure Technologies for about 26 years, which is a company that specializes in oral fluid drug testing. And so for the major part of that time at OraSure, I was involved in the launch of oral fluid drug testing products, and also helped kind of along the way with helping with data for guidelines and things that have now turned into SAMHSA and DOT, regulated testing guidelines. So I really enjoy this particular topic because I think it's so powerful and has so much great application in the field of drug testing, especially for workplace. And so I'm rather passionate about it. I'm sure you'll hear that in my the way we discuss oral fluid drug testing.
Yeah, and this is one in a series of podcasts on oral fluid drug testing. And we're going to start with some basic items. We find that a lot of people don't realize that there's a difference between oral fluid and saliva. Maybe you can educate us on that.
Well, sure. So saliva is really a fluid that we all have, we make every day. And trivia fact, we make between 1 and 2 liters of saliva every day. So there's an abundant quantity of it. If anybody was concerned that there isn't enough, that's really not the case. And it's really a saliva comes from several major and minor glands that you find in the salivary areas of the mouth, and it can be generated just naturally, right, by the way when we eat or when we do things, we generate saliva. It actually can also be generated mechanically. There are certain devices that can be used to pull out saliva. And typically it's what you would collect if you were to spit into a cup or into a vial. And there are some tests on the market that act that way or have that sort of collection method. And then oral fluid is a little different. It's actually the fluid that you collect from the mouth, typically where you take a pad and have that on a stick. And in a future episode, we'll talk about some of the devices more specifically. But basically the pad goes into the mouth, either between the cheek and gum or under the tongue, and it's able to draw out sort of a material that's a little bit more serumy and a little bit more like what comes from the blood. And so that has historically been used for things like large proteins, HIV, things of that nature. But as we've arrived kind of at a time where I think oral fluid has become more of the term that people are using either in guidelines or in the market and different products, there shouldn't be too much concern about whether you're talking about saliva or oral fluid. The terms are kind of been used intermittently, and that's fine. The more important thing is that the developers of the tests and the things that we're doing in the field from an industry standpoint are developing those the cutoffs and things we're going to talk about next that are suited for the different sample types, and that basically you're able to hit the cutoffs and you're able to perform in those samples, whether you call it saliva or oral fluid, doesn't really matter. It has to basically meet the standards that are being set for the industry today.
So for many years, we heard people refer to saliva alcohol tests. Mm-hmm. Is that the same concept?
It really is. For the saliva alcohol test, for example, the QED one or the there's a few other products on the market that really is swabbing with a stick that has a swab on the end, and you're swabbing the mouth. You're collecting saliva, probably also some oral fluid in the swabbing process. The combination of them, which then goes into the saliva test for measuring alcohol content in that sample.
So going back to drugs then, how do drugs get into our oral fluid? Why are they detectable in an oral fluid sample?
Right. Well, essentially, they come from the bloodstream, Bill. So the person that's using a substance, whether they're smoking it or they're snorting it or whatever methods they use to take that substance, it goes into the bloodstream. And once it gets into the bloodstream, the process begins where it starts to cross over into using the terms I used before, the salivary glands, right? Those are in contact with the bloodstream, so they feed the mouth through those glands, and so the drugs and other things come into oral fluid, and that's why we can test for them. They're there, and they're there at lower concentrations, and we'll talk about that a little bit when we talk about cutoffs and the differences between some of the sample types. But they're there at a concentration that's plenty good enough for us to be able to screen for them and do confirmation testing and all the things that we need to do. One of the things that's a little different is that with saliva and what comes into the saliva from the bloodstream, it tends to be more of the parent drug, and less on the metabolites. And that's because the body hasn't really broken down that drug to any large extent. Like what you find in urine, you find a lot of the metabolite primarily. You won't find parent drug to any significant extent or any extent at all. So really what you're testing for in oral fluid tends to be more of the parent and some metabolites depending upon the drug and some of the basic science and chemistry, which we won't get into here.
So for many, many years, I mean, going back into the 90s when drug testing was kind of sort of still in its infancy in terms of workplace drug testing, you often heard some, really smart people in the industry refer to blood as sort of the gold standard, and that oral fluid sort of mimicked blood. Is there a correlation there?
So yeah, there is, because again that you can look at the oral fluid as a filtrate of blood, and there is a correlation between the concentration in blood and the concentration in oral fluid. It's not exactly a one-to-one correlation though, and drugs come into the oral fluid in a different way for each drug class, and it has to do with what we call the pKa, or the acidity or the alkalinity of a particular drug, and the environment that it's in will determine to what extent that ratio, you might call it plasma or oral fluid to blood ratio or oral fluid to plasma ratio, what that number will actually be. But again, at the end of the day, it doesn't really matter how well it correlates. What really matters is that when the test is developed for the oral fluid sample type, and someone is aiming for a certain cutoff with an assay, that you're able to pick up that drug in that sample, and you're able to effectively determine whether something should be called negative or called positive.
So I know we're going to cover this in more detail in a future episode, but when you say detecting the drug, you're talking about the drug itself, sometimes referred to as the parent drug, as opposed to a metabolite of the drug.
Yeah, it's really both, but primarily we're testing for parent drug. THC is a very good example, marijuana, and we're going to talk about this one as well. It's such a hot topic today, of course. In oral fluid, you won't find any of the metabolite, at least not with common methods that are used today. You'd have to look very, very hard, and it would be at extremely low concentrations if you even did find the metabolite. You're going to be looking for primarily parent drug with THC. Some of the other ones, there's a little bit of metabolite in there as well, depending upon the drug. And the assay that's developed to detect that drug, it might be a class assay. Like cocaine, for example, you're testing for the parent. A lot of that comes into the oral fluid, but most of the tests that are out there for cocaine are also picking up benzoylecgonine, which is the primary metabolite of cocaine, because that does make its way into the oral fluid sample.
So maybe for our audience today, just give us a quick definition of the word assay.
Sure. Think of it as a test. The things that you might be familiar with over the counter, buying a test, for pregnancy, or buying a test for COVID or whatever. An assay is really you're testing for a particular substance. Could be a drug, it could be infectious disease like HIV, it could be something else. But an assay really means a test. And furthermore, we'll get specifically more into this, but the term immunoassay comes into play in our field, because these tests or these assays utilize immunology to come to a result. And what I mean by that is we're all familiar with antibodies, right? We make them in response to antigens or viruses or things that come into our system. And so those antibodies are able to be used in laboratory tests or assays. And it's quite, fascinating actually, because in the laboratory, you can make these antibodies that are specific for THC or for cocaine or for methamphetamine. And you do that either through animals. We might call that a polyclonal antibody, where it's done in a sheep or a goat, or monoclonals, which are made in mice. And either way, you're basically synthesizing an antibody that has, let's say, if you're thinking about a lock and key, they fit together. So if you've got a THC antibody, you're able to then use that in an assay that can detect the molecule of THC. And that's basically how these immunoassays are developed. And you use other things that become your signal generator, right? Whether it's a colloidal gold particle in a rapid test, or whether it's an enzyme that's used to generate color in an immunoassay that may be used in a rapid test or in a laboratory-based test. Probably a lot more than you wanted to know, but that's the definition there.
But the bottom line is that in an oral fluid sample, many drugs can be detectable.
Correct.
And the laboratory would need an assay for each of those drugs.
So yes, a laboratory, when they get a sample, they only need one oral fluid sample, but they need multiple assays in the laboratory. And these get run on instruments. So you'd have an assay for cocaine and one for methamphetamine and one for THC, and et cetera, et cetera. So those would all be put onto an instrument that's able to run that one oral fluid sample and multiple drugs all at the same time. And so these are called automated immunoassay analyzers.
So, final question then.
Yeah.
If you're in a state that requires an employer who wants to do oral fluid testing to use an FDA cleared device, would we be talking about an FDA cleared collection device and FDA cleared assays for specific drugs?
So yes and no. So if you are required to use an FDA cleared test and you're doing a rapid test, that rapid test device has to carry its own FDA clearance, if you will. We call it a 510K, right? So there's no way around that. That whole test cassette or device has to be FDA cleared. If you're to be using a laboratory-based test in a state that requires your test to be FDA cleared, then traditionally the device, the oral fluid collection device that you're using to collect the sample and the assays would be FDA cleared together. And that's how historically it has worked. Now, today, here we are many years later from when the FDA has set those sort of guidance. There is room today for a manufacturer and for laboratories to be able to use their assays in the lab that are lab validated and developed and run them by mass spec, which is getting pretty far down the road in terms of the advanced level stuff. But those instruments can be used without being FDA cleared. As long as the device you're using is FDA cleared, it can be used with these alternative immunoassay or alternative testing technologies in a laboratory. But by and large, the standard pretty much is and will continue to be, especially for regulated testing, FDA clearing the assays and the device to be used for drug testing, particularly for DOT and for regulated testing.
Perfect, Brian. Thank you very much. And I know we wandered a little off the path of the sort of beginner topics, but it's all sort of very connected. And as one listens to an explanation of basic things, their minds will obviously wander off to more advanced topics and questions. So this was excellent. We're going to end today's podcast on that note, but we encourage you to check back with us on a regular basis. Go to our website and look for additional podcasts on oral fluid testing and many other subjects under the currently speaking section of our website where you'll find all of our podcast recordings. Brian, thank you very much for joining us today.
Absolutely. My pleasure.
Bye, everyone.